Just a little bit of introduction about myself. As you heard, my name's Helen Kalirai. I'm a postdoctoral scientist at the University of Liverpool. I work with Professor Sarah Coupland, who some of you may know, and I'm also closely linked with the Liverpool Ocular Oncology Centre because I actually manage the day-to-day running of the molecular genetics service that's undertaken for the patients. Before I sort of delve into the UMCure2020 project, it's just to say there will be time for questions at the end. However, if perhaps you think of questions after that, or there's something that you don't have time to ask at the end, then there will be a question box, which Andre Valente is going to point out now. And we ask that you put any questions or thoughts and views in that box, and hopefully we'll be able to address them at a later time. And also, tomorrow, for those of you who are attending the European Melanoma Patient Network meeting, both Andre and Dr. Manuel Rodriguez from the Institut Curie at Paris will have a session there, and the session will be interactive, so you'll have lots of opportunities to chat about things then as well. So, we were very fortunate before Brexit, because I know it's going to come up, to actually be able to participate in Horizon 2020. Now, if you've not heard of Horizon 2020, it's an EU research and innovation programme, and it's particularly good because it brings together, the idea is that it'll bring together European partners who are partners of excellence in their field. And that by doing this, we won't have unfocused research like you heard about before lunch, that actually we will have a very focused programme of research. And this will help us tackle those societal challenges that we know are out there. So there's approximately 80 billion euros of funding available through Horizon 2020 over 7 years, so between 2014 and 2020. And in 2014, a rare disease call, so the Horizon 2020 put out calls for applications. A rare disease call was put out for applications focusing on the development of new treatments for rare diseases. And as we all know, this is very important for patients with uveal melanoma. And so Professor Sergio Roman at the Institut Curie had the foresight to bring together a panel, a consortium of European partners, and with the help of a project management company called Seeding Science, an application was submitted. And we were lucky. We were one of 10 new research projects that were funded as part of this call, and our project started in January this year. Okay. So as I said, UMCure2020 is a consortium of European experts, both at the clinical ocular oncology level, basic scientific research level. We're hoping to, well, we are working with patient organisations, and the aim of the project is to develop new therapeutic approaches to treat metastatic uveal melanoma patients, but also to empower you as patients and to share this knowledge efficiently. So I want to just spend a little bit of time introducing the partners so you know who is involved in this project. So there's 12 European partners. So 4 of these are major referral centres, so centres that may also be research institutes that see a large number of patients with uveal melanoma. So this is the Leiden University Medical Centre in the Netherlands, Institut Curie in Paris, in France, the University of Liverpool, as I say, works closely with the Royal Liverpool University Hospital, and the Jagiellonia University in Krakow in Poland. There's 3 other research institutes that are involved, the Cancer Research UK Manchester Institute under the guidance of Professor Richard Murray, the University of Trento in Italy, and the University of Leiden again in the Netherlands. We have 2 small biotech companies who are involved, PEP Therapy and Pamgene, and I'll talk about them in a little bit more detail when I go through the work packages. We have the Champillimo Foundation, of which Andre is the driving partner for that. The Champillimo Foundation has a strong record in patient-centred care, particularly cancer patient care, running clinical trials, patient advocacy, and those, the important things that we need to bear in mind when we're thinking about the work that we're doing. We have the project management company, Seeding Science, who will help us all the way throughout the project and make sure that we meet our targets and deliverables. And also, I've already mentioned, we have an advocate, Bettina Rill, from the European Melanoma Patient Network. So the aim of the project is to make it a patient-centered project. And so when we look at how the project's been designed, it's been designed with the patients at the center. And the way that the project will work is it goes through a series of work packages, and I'll go through each one of these in briefly, not in great detail, individually. But the aim is that it's a continuum. So the basic science that's being undertaken within these work packages that are being driven by patient samples will eventually lead all the way back to the initiation of clinical trials and back to you as patients. So, as I say, I'll spend a little bit of time going through the work packages so that you can see what they involve and what we're trying to achieve. So, work package 1 is actually key to a lot of the downstream packages. So, work package 1 aims to establish a European uveal melanoma biobank network. And we already have the 4 uveal melanoma referral centres, as I've outlined before. Now, 3 of these, the top 3 here, so Liverpool, Paris and Leiden, have established biobanks already. So we're already collecting biosamples from patients who attend the clinics at these referral centres. The third institute, the Jagiellonian Institute in Krakow, will use the well-established protocols and knowledge that we've already gleaned over the years, if you like, setting up these biobanks to contribute and set up their own biobank and feed in samples into the various work packages of the project. So there's a number of tasks and deliverables that we have to achieve with the biobank. The first one of these was to agree on minimal data sets that needed to be available for each patient sample. And these include clinical information, histopathological information, and genetic information. And so we've agreed on these data sets already. And so that's now allowing us to extract data from each of the centres' databases. So the 3 centres where we already have patient samples, we will be extracting data from some of those samples where the patient consent allows us to use the samples in this type of project. And that's very important because we wouldn't do anything with the samples without the patient consent and the appropriate ethics. So I should point out that although we do have these 3 established biobanks, when you look at the material that's within them, the majority of the material that's held in the biobanks is either primary uveal melanoma material or it's fixed material. And that's very important when we look at the downstream packages, because what's really unique to this project is that we're going to look at metastatic uveal melanoma material. So there's a lot of work that's gone on over the years on the primary cancers, but really we need to know how similar are the metastases to the primary tumours, where the differences lie, and then use that information accordingly to inform the clinical trials that we may want to initiate downstream. So there will be a prospective collection that will be ongoing at the 4 centres. And in order for us to do this properly and efficiently, we have to create harmonized procedures. So at each centre, we will be using the same procedures, or as close as possible, for collection, storage, transport, processing, anything that we do with these samples, so that if a sample from Institute Curie needs to be analysed by a particular method, then the same sample in Liverpool can be analysed by the same method because it's being collected in the same way. So that we're minimising any, I suppose, inter-institutional variability. So in work package 2, we will be testing, we will be performing the preclinical validation of biology-driven therapeutic approaches. So this will start out with single drugs, drug combinations, And these will be tested initially in a panel of cell lines, and across the institutes we have quite a panel of these cell lines that reflect the various differences, either genetically or morphologically, between what we see in patient samples. So there'll be a triage, because there'll be a lot of agents tested initially. They will be either repurposed agents that are already FDA approved, or there'll be novel agents. So we will triage them initially in the cell lines, and then they will move into more complex models. So we have already at the Institute Curie, we have what we call patient-derived xenografts. And these are models in which tumour fragments from patients are implanted into mice. And there is strong evidence from the work that they've done that they are highly reproducible of how the patients were in the tumour, and we'll also how the patient tumours looked when they were removed. And we will also use a zebrafish model as well. So this is going to be conducted in 2 phases. The first phase will use current disease knowledge, what we know about signaling pathways that are disrupted, other information that's out there in the literature, and we'll also use the available models that we have to us now. So there is a panel of PDX models at Institute Curie, but we hope to roll out this model to some of the other research institutes in the consortium. In phase 2, we will identify new targets, and I'll discuss, this will be based on information that we obtain from Work Package 4, which I'll go on to discuss. And we'll also, hopefully, over the course of the project, develop new disease models that perhaps better represent the individual disease. So throughout this work package, we'll also be collaborating with PEP Therapy. Now, PEP Therapy is a small biotech company that develops innovative cell-penetrating peptides. And the idea is that these peptides can inhibit protein-protein interactions. So in the signaling pathways that you've seen on some of the slides earlier this morning, there's a number of factors in those pathways, and those factors interact. And if we can stop them from interacting, then this is one approach that this company is taking. So as I've already briefly mentioned, Work Package 3 looks at the development of the next generation of preclinical models. And so we're going to further develop what we already have available and new models. So the new models will be things like genetically engineered mouse models. So I've discussed patient-derived xenograft models, but we also have circulating tumour cell-derived xenograft models as well. But I won't go into great detail on that. So Work Package 4 will rely on the prospective collection of samples that is coming into the biobanks. And this is also really key to the project. So we will characterize these metastatic samples as well as feeding them into the preclinical models. We will use some of the samples for in-depth characterization. And the idea then is that we will have a greater understanding of the molecular changes in the metastatic uveal melanoma, and that will allow us to generate new target hypotheses for Work Package 2. So one of these approaches will be to look at the genetic changes in the metastatic samples and really go to town, really look deeply at the genetic alterations that we're seeing. And because it's a consortium and because we're able to bring in samples from the 4 referral centres, we'll have a larger panel of samples. We'll be able to look at things like tumour heterogeneity between patients. We'll be able to look at the things that we heard about this morning, with the sense that one patient may have an SF3B1 mutation and therefore respond differently to drug A as opposed to, patient B who may have a BAP1 mutation and therefore could respond to a different treatment. We'll also be looking at the signaling pathways in these tumours. So this is in collaboration with PAMgene, who have great experience in conducting proteomic analyses, and in particular something called phosphoproteomics. They will be helping to develop peptide arrays so that we can do this more quickly. And again, this will all feed in together with the genetic information to provide us with hypotheses. And finally, we'll be looking at the characteristics of the immune landscape in the liver it will be predominantly liver metastases. So this will be looking at things like the macrophage infiltrate, T cells, the other immune cells that are infiltrating the tumour. And this really hasn't been done in any great detail before. So, you know, again, this is key to our understanding of how immunotherapy may work, or maybe alternative therapies together with our partners, including PAM-gene and PEP therapy. And in work package 5, this will really be going on throughout the study. And this is very important because it's me being here today, it's the widespread dissemination and exploitation of what we're doing. You know, presenting the results, not only at scientific conferences, but to patient groups, to hearing what you want us to achieve throughout this project, to increase the information that you receive and your involvement, to generate a dedicated uveal melanoma patient and caregivers portal, as well as a project website, and also to try and establish European-wide initiatives for patients to meet and to network and to get involved. And this is, again, as I mentioned before, where the Champilamo Foundation, who's located in Lisbon in Portugal, is key to this work package. And hopefully Andre will have a great interactive session with you all tomorrow and be able to listen to what you've got to say and how that can help us in this process. So we've gone some way to starting creating these interactions in Work Package 5. So we have the UMCure2020 website, which I would recommend that you all have a look at. You give us feedback. We've also presented this work at a cancer research conference, the European Association for Cancer Research in Manchester in July. And we'll also be presenting it at the EVA meeting in Nice in October. And Bettina will also be here as part of the European Melanoma Patient Network tomorrow. And so again, I'd just like to reiterate, please post questions and suggestions in our box outside, and they can be discussed at the interactive session tomorrow. So I'd just like to finish by saying thank you for listening. I'll try and answer any questions. I'm not involved in all the work packages, but I'll do my best. And yeah, thank you very much.
Anybody have any questions for Helen? Don't forget, she said as well, to write them down and put them in the box if you think of anything else. I do actually have a question.
I've got one.
Oh, hold on, Dick.
I'm from the Netherlands. You were speaking about the European Biobank network, which is already starting up. What's about the EU regulations about sharing data? Because as we speak at this moment in Brussels, they're gonna make new laws, which makes it more difficult to share data.
Okay, so all the data will be anonymized or pseudo-anonymized, so there won't actually be any patient information that is transferred into the virtual database. So what we do when we have our patient samples is we code them.
Hi, Helen. I wanted to ask, the fact that this is due to finish in 2020 and it's only 4 years away, and there's a lot of research at a lot of centres. I mean, how confident are you that these will be seen through and that we'll get to the later stages of clinical trial development as a result of the research that itself has to take place? And what controls and handover have you got at the end of that, perhaps?
Yeah, so I mean, when Horizon 2020 look at the applications, they obviously do consider that, and we have to put in deliverables and milestones. So we have to achieve those deliverables and milestones at specific time points all along the way. If we don't achieve them, then the money will be withdrawn. So we do have a lot of confidence in what we've put forward that we will be able to achieve. And I think the fact that we've got so many European centres who are already working on this and already have the expertise. We're not trying to develop something that's not already in place. We're just trying to expand on that, really, rather than creating something new.
If it's okay, I'll ask my question. Okay. I was thinking about something that both Serge and Amin picked up a little bit on, that some patients with metastatic uveal melanoma seem to have a more slow-growing disease where perhaps about 10% may be able to have a resection. And I'm wondering if your research will incorporate apportioning out those metastatic tumours that you're assaying and doing all that with, so that we can see if there's any difference between those two types of patient. Does that make sense?
Not quite.
So I'm wondering if they're biologically different.
Yeah,
Does that make sense to you, Serge?
Yeah,
yes.
It makes sense to me, but so we obviously have the patients where we can get specimens because they are resectable. Are you saying that they're the more slow growing?
Well, Serge was talking about how, and actually Dr. Karidis, about doing laparoscopic. A lot of places do the laparoscopic intervention. They can obviously take a biopsy at that point.
Yeah, okay. So we are hoping to get the laparoscopic biopsies as well. So they're, as part of the CellPack clinical trial, we've, if you like, tested the water about how the hospitals and the patients feel about us perhaps taking an additional biopsy on top of the diagnostic biopsy. And it seems that the majority of patients, because it's not seen to be associated with an increased risk, are quite happy for that to happen. So yes, we will be trying to look at exactly that and collecting the clinical information that goes with it. So whether they've got miliary disease, if it's a single large deposit, I mean, that is very important because I would imagine that the tumours could be very, very different depending on, yeah.
Thanks ever so much, Helen.
Thank you.